Nearly all of our cells share the exact same genetic code, because with few exceptions, all of our cells have the same DNA. So how does this neuronal cell look and function differently to this funny looking immune cell? Welcome to the last generation to age in our series on the hallmarks of aging. In this episode, we'll cover epigenetic alteration. Now that might sound complicated, but we can break it down further. In our genomic instability episode, we said the genome was like a master blueprint for building and maintaining all of our cells. Imagine a master blueprint for building every building in the world. If we tried to build straight from this blueprint, we'd end up building a mess. So we need some instructions telling our cells what to build and what not to build. Our cells do this by turning genes on and off, like switches. Each unique combination of on and off switches equals a unique cellular identity with its own appearance and function. Now, to understand how our cells actually accomplish this, we need to first understand how our DNA is packaged and then expressed. Our DNA wraps around proteins called histones, which coil up the DNA, packaging it into bundles called chromatin. When chromatin is coiled tightly around many histones, chromosomes are formed. One way we can turn genes off is by attaching chemical groups to histones, such as methyl groups. This makes the histone coil tightly, hiding the DNA from transcription enzymes. Alternatively, we can remove the methyl group, opening the gene back up for transcription. Another mechanism of controlling these switches is by adding chemical groups directly to the DNA itself. For example, if we add methyl groups directly to the DNA, will prevent the binding of enzymes and therefore shut down gene expression. All of these mechanisms and more are what's known as epigenetics, which literally means on top of the genome. The thing is, these switches that we add on top of the genome, they become less precise over time, leading to a loss of cell identity. This leads to many of the consequences we've discussed before, such as cellular senescence or zombie cells, or even cellular proliferation leading to cancer. The good news is the loss of these epigenetic signatures over time can be tracked and correlated with chronological age, giving us biological clocks. And what's more is epigenetics seems to be a reversible hallmark of aging, but this we'll cover on later episodes. Thank you.